So far we have only seen the bacterium's outer raincoat, the Glycocalyx. But if we peel that layer away, what do we find next? It's the bacterial cell wall, the structure that is going to give the bacteria their shape and protect them from the bursting. Let's go one layer deeper. Imagine guys, you are trying to fill a water balloon with more and more water. What happens if the balloon has no support? It is going to get burst. Same way, if a bacterial cell faces a similar challenge, like water is constantly trying to move into the cell due to the osmotic pressure, without a strong outer wall, the bacterium would swell up and burst. That's why the cell wall is so essential for the bacteria. Think of it as a strong brick wall of a house. It does not make the house alive, but it's going to give the house its shape and protect everything from the inside. Like, what is the composition of the bacterial cell wall? Actually, it is mainly made up of a unique material called the peptidoglycan, which we also call it as a murin. It's a alternate terms, guys. Peptidoglycan is also called as a murin. Here is an exam point. Like, plants have cell wall as a cellulose, fungi have chitin, but bacteria have the peptidoglycan. Never mix this up. Now, what is the function of it? So what does the cell wall actually going to do? It has a 4 main important jobs, guys, where this cell wall is going to give the bacterium its shape, and it is also going to protect it from the mechanical damage and helps in preventing the osmotic bursting and help the bacteria survive in the different type of environment. Now here is something that scares many students: gram positive and the gram negative bacteria. But trust me guys, it's much simpler than it sounds. Imagine you are wearing a winter clothes. One person wears a very thick jacket, another person wears a thin jacket, but also carries an extra raincoat, right? Which one has the thicker clothing? The first one. That exactly the difference. Like gram positive bacteria, they have a thick peptidoglycans as a cell wall. Because of this, they retain the purple gram stain and appear the violet in color under the microscope. Gram negative bacteria next, they have a thin peptidoglycan layer, but outside it they are going to possess and one more additional outer membrane. Because of this extra membrane, they do not retain the purple stain. Instead, they are going to appear pink after the gram staining procedure. Quick memory trick guys, think of it like this: positive means plenty of peptidoglycan, negative means your outer membrane is present. It's not the official definition guys, but it helps you remember during the exams. So we have crossed the strong protective wall, which is cell wall. Now we are going to enter finally into the living part of the bacterial cell, that is the plasma membrane. It is the boundary between the inside of the bacterium and the outside wall. Imagine guys, the security like a security gate of a high-tech company. Not everyone is going to allow to enter, right? And not everything is going to allow to leave outside. The security gate decides similarly. The plasma membrane controls what should enter and what should exit the bacterial cell. Now, what is the structure of plasma membrane? It is mainly made up of phospholipids and the protein. It follows the fluid mosaic model, meaning that the membrane isn't rigid. It behaves like a flexible moving sheet. So what are the functions of plasma membrane? The plasma membrane performs several important functions. The first function is selective transport of the substances. Second is cellular respiration in bacteria. Third is ATP production. Fourth is secretion of enzymes. Remember this carefully. Unlike eukaryotic cells, bacteria do not have the mitochondria. So where does this respiration occurs then? It is going to occur on the plasma membrane. This is one of the most frequently asked concept in the biology exams. Now look closely at the plasma membrane. Do you notice those inward folds? These folds are called the mesosomes. Think of them like folding of a sheet of a paper. The paper doesn't become bigger, right? But folding creates more usable surfaces. Similarly, these inward folds are going to increase the membrane surface area. Like mesosomes have been described in our textbooks, like helping in cell division, like in DNA replication, respiration, and for enzyme secretion. However, one interesting fact is that many modern researchers believe that mesosomes are observed in the older electron microscopy studies may actually be artifacts like formed during the sample preparation. Even so, many university and the competitive exam syllabus still include mesosomes, so study them according to your syllabus. So far we have traveled from outside of the bacteria to the plasma membrane. In the next part, we'll enter the cytoplasm and explore the bacterial control center, the nucleoid, along with the plasmids, ribosomes, and the inclusion bodies. That's where the real machinery of the bacterial cell is going to begin.